introduction session on current trends of medicinal chemistry, applications for the drug developments. I request all of you to please be seated and occupy the front seats first. Thank you. The title of today's talk that will start soon is General Skills in Medicinal Chemistry, an introduction session.
Okay. Thank you very much. So, try which once more. So, a warm welcome from my side to all of you. It's a great pleasure and also a great honor being here. And we will start slowly and then slowly increase the pace for our learning here in this area. Since chemistry is a lot about networking and so on, I will start showing a little bit what we are doing and where you can interact with us or where some other possibilities are with us. Then I will this extend to Germany, in which way Germany has some things to offer, especially for very talented organic chemists, medicine chemists, pharmacists in this way. And then we go into some starting material, what skills you should have and in which way you should start. First of all, Düsseldorf, Germany is far away, but like Nagpur, which is in the center of India, Düsseldorf is the center of Europe, more or less, in the way. And thereby, you can see Düsseldorf, we do have a driving distance to all big cities in Europe, and also by flight, within a short time, you can reach all other areas into these different things to go there, and so on. And for Düsseldorf, it's also quite important. It's the capital city of the federal state North Rhine-Westphalia, a country state of Germany. And it is localized with the Rhine River, so very prominent, very nice area to stay. And we have a cold winter and a warm summer, but warm summer is less warm than your winter, I have to say. So that's something in between for the different areas. So what you see here, especially now, this Rhine promenade, where actually now a lot of people really are in this different areas. You see, we have a presenting house in a city, but the university here called to the poet Heinrich Heine, a famous German poet in this way into this area, and we are presenting here all the different areas. And maybe you see here, this is one of our lecture room in the medicine sector. You see some fine arts there, and this is a original picture by Andy Warhol, who spent some time in Düsseldorf. And Düsseldorf is very famous also for its fine arts, modern fine arts like Beuys or Gerhard Richter. They have their origin from Düsseldorf and stay still in Düsseldorf if they're still alive. Another important point in Düsseldorf is that we do not have summer, autumn, winter, and spring, but in fact we really have a fifth season in there. And today is the high point of this fifth season because we have and the carnival today we have the Monday's Rosen. This is exactly today, where a lot of people are going outside and have it. I think it's a little bit different to the Rio Carnival, but nevertheless it's worth being there, and thereby I skipped this event just for be with you here in this area. So thereby Carnival is quite important. It has a good point of view. Nevertheless, we are not only celebrating Carnival, we are also making a lot of compounds, and there I have different topics where we are and where we are working on this. So you see, we are. This is a phylogenetic tree of some G protein coupled receptors, and you see that we are working in the histamine receptor field and in the dopamine receptor field. This is our main task, and thereby there are different receptor subtypes, and we are not working on all receptor subtypes simultaneously, but we focus on one or the other. So we mainly focus on histamine H3 and H4 receptor, and we mainly focus on dopamine D2 and D3 receptor, and so from time to time we could not leave the other receptor out, but they are not our main focus in the different areas, and there are some compounds which are strictly available. So there we are working on some G protein coupled receptors, and dopamine and histamine are our main purpose on this. But you will see, or not see really, that we are working in the lipid area, in the sphingolipid area, and in the arachidonic pathway. So with the biogenic amines, we are in the very hydrophilic area, and with the lipids, with the sphingolipids and the arachidonic pathway, we are in the lipid area. So we extend our knowledge. All of this belongs to receptors and enzymes, but here we work on glutamate receptors. So NMDA receptor is a subtype of the glutamate receptor, so we also work on different channels in this way. There we do not have our own screening system, but nevertheless we do have some cooperation partners, and we are developing these compounds. So you see what we are doing is we are playing with small compounds. We are juggling in this small compound, and that's our main field. We are synthesizing the compounds, we are purifying and analyzing these compounds, and also make the characterization due to their pharmacological profile. This is everything what we are doing in-house, really, and then we go with additional screenings outside with cooperation partners in this way. And you see here with the biogenic amines like dopamine and histamine, with ion channels like NMDA receptor, N-methyl D-aspartate receptors, with the sphingolipids and also with the arachidonic pathway, then we change a little bit from the hydrophilic to lipophilic area in the different pockets this way. I will not speak a lot about our lipid area because I will not spread all our information on this and would focus mostly on the histamine receptor and histamine receptor subtype because the most successful area in this way. But nevertheless, I would shortly enhance that we are working on the arachidonic pathway, and you all know acetylsalicylic acid, which is a cyclooxygenase inhibitor, the COX inhibitor, and they are working on this pathway for the arachidonic pathway, inhibiting the synthesis of prostaglandins. And there we do have some new compounds developed, which they are developing cyclooxygenase, 5-lipoxygenase, but also soluble epoxyhydrolases, so different enzymes responsible for inflammatory processes and pain processes in the way. And there are also some new targets called the resolvins and so on, which are anti-inflammatory or resolving mechanism in the different areas. And there are some new targets even in this very old field of the arachidonic pathway in this way. A little bit newer are the sphingolipids in this way. They are mainly localized in the central nervous system. So there is responsible for neuronal processes in this way, and this is then activated swinging to swing, and then transported outside and can then act with different sphingosine receptor subtypes in this way. There we are not only working on the receptor subtypes, we also work on the regulation of the different processes. So the transporter system is one of these areas, and we also look on the phosphorylation, the activation of sphingosine to sphingosine phosphate, the active ingredient in this way, or regulated by ceramide synthesis. Thereby we also work on ceramide synthesis in this way. So you see that not only on receptor, also on enzymes, and sometimes the anabolic and catabolic enzymes are quite important on the regulation process for the different structures. What I would like to stress here is not that you go with me into the details of the sphingolipids or different areas, but that you see that there's, if you look on the target, there's more than one control for one target. And you have to look not only on your target in which way your drug is interacting with the target, but on the control mechanism. Because we have a living organism, the cell is always in reaction to anything what's happening there, and thereby you can have receptor upregulation, downregulation, different coupling. The transmitter is fastly or is not used in a different way, that you're not looking only on your target, also on the surrounding and so on. And the better you have then drug development, the more surrounding on the different areas. So thereby not focus really only on your target, but then on a different way. Then what we are doing, once more, you see here the NMDA receptor, the histamine receptor, the dopamine receptor, and the different lipid receptors in this way. And this is our target, what we are working on. With the targets we are working on are connected to different diseases in this way. So we can stimulate or inhibit the different targets, and thereby can manipulate different diseases in different stages. You see here schizophrenia, epilepsy, morbus Parkinson, sleep disorders, Alzheimer, multiple sclerosis, and so on. So there are different diseases in this way. They are interacting in different points. The reason why we have chosen the different targets is that they are connected to these diseases. They are not only monofunctional, but they are connected to each other. So we understand the disease better, and we know many of the targets connected to the disease. And that's the reason why we're getting some, at a certain level, some expert, not only expert for the target, but also expert for different diseases. Not with all targets, not with all approaches and so on, but thereby we are not focusing on all diseases, but we are focusing on some diseases connected to our targets and so on. So thereby some phenotypical structure and some target-oriented structure into this way for the different structures. And then we go, when we hit the target, we have different possibilities to hit the target. The histamine receptor is, for example, 4 different receptor subtypes. Dopamine is 5 receptor subtypes in the way with different function and so on. So we are addressing the different receptor subtypes to a different approach for the different structures in this way. And there we cannot take all of this structure, because when you look for serotonin, for example, you have 17 different subtypes in this way, and then you get out of order with the different structures. So we are only focusing on some limited to our working progress on what we can do. So we're looking here on the receptors, but we are also looking for the anabolic and the catabolic enzymes connected to this, so the surrounding connected to this area in the way and so on. Yeah, and you will see my last speak will then on a multi-targeting compound on Friday, and then you see that we are addressing not receptors, also addressing the enzymes connected to these structures. And that maybe then gives some additional properties, for example, in different areas, but also some additional complexity, and complexity always gives additional problems. So that's always the case in the different areas. And to add complexity to targets, I would also add complexity to the effect of this structure. So we can address the target, and we can make some agonist behavior, or we can make some antagonist behavior, or we can have some partial agonist behavior in the different areas. So different efficacies are connected to the target. So not only the affinity is important, also the efficacy is important and so on. And thereby we can have selective compounds acting on more targets, but having a targeted efficacy level. Agonist at one compound or one target, antagonist at another target and so on. And thereby we received targeted selectivity for the different structures in this way. Yeah? And thereby these properties are quite important for different structures, and we will go on with this in a quite clear way in this way. Nevertheless, once more, I will not show everything what we have done. I will focus on the magnificent 7, the 7 transmembrane receptor systems, the G protein coupled receptors. And there I would mainly focus on the G protein coupled receptors and not on the G protein, which is already another target. The heterotrimeric target, and other groups are handling with the G protein. We are handling with the membrane-bound structure there. And we are exploring the space here, and thereby mostly with the aminergic receptor like histamine and dopamine, like I explained in the different ways. Yeah? So what we are doing, it's different to many other people. We are making only small molecules, and we are characterizing only small molecules. This is our expertise. We are not going into biologicals or protein structure, larger structure, and so on. So we keep to our expertise in this way. And this, from time to time, we are asked we can do something other, but our expertise lies here, and we stay to our expertise. We expand our expertise. We take new demands, new tasks, but not beyond. So you must be aware what you can do or what you cannot do in this way. Yeah? And you see, these pictures are just that we are handling with the different structures. What we can do is a small molecule chemistry. We can build up different compounds. And I explained already to the students that it's not important in medicine chemistry that you have a very efficient 24-step stereoselective synthesis in medicine chemistry. This is very fine in organic chemistry to have. Yeah? So you have three generations of PhD students working on this synthesis route, and then you can describe this step by step in this way. In medicine chemistry, it's important to have the final compound and to have enough final compound with the characterization and so on. So if you have a one or two step reaction leading directly to your compound, that's fine. Yeah? Then you can build up good structure-activity relationship and things like this, because you can do much more variations and so on, and you do not prepare only one compound for the whole time in this way. So thereby, the synthesis must be efficient, yeah? But you must also be aware if you need a stereocenter in this specific compound, or you need to explore this stereospecific center in this way, you must be competent enough to prepare these compounds. So high skills in medicine, in chemistry are needed in certain cases. Not always, but in certain cases. So sometimes you need this 24-step stereoselective synthesis. Yeah? Then you have to be able to do this. Yeah? Or to partner up with a different structure. Yeah? Then you see here chemistry is one important point, structural biology. Know your target, know the structure of the target, know the interaction. We do have molecular biology, know the signaling mechanism, in which organisms the signaling mechanism take part, where is this target co-localized and so on. So know the physiology and pathophysiology of your target and so on. And then know the pharmacology of this. Especially when you develop a new drug, and a new drug for maybe for a disease which high medical need and so on, this target has not been approached so far, and is not known what effects are really connected to this drug in the whole body at the end. Maybe you know the effect in liver tissue, yeah? But that doesn't give you any information what its effect in the brain, for example. So thereby, that is quite important to know into the different areas. So this is more or less what we are doing in-house. We are doing the synthesis. We are partnering up with virtual screening, so molecular modeling and so on. There are some experts, and we are not the experts in this, so we are connecting to the experts in this field, and then we take their advice for new synthesis route and so on. So thereby, we take them for lead optimization and so on, but we are doing the synthesis, purification, analytic, and some early biological in vitro screening and so on. So that's also important for us that we have the in vitro screening in-house, that we can easily coordinate the round, that the PhD students with the synthesis got the information this compound is active or not active, then make some modification in their synthesis route or their structural design in this way. So we cannot wait always for outside partners to give us the results. It's better to have them inside. We cannot do everything, so we also need these outside results, but for early screening and for early orientation for drug design, it's really helpful and, to my mind, really essential to have this data really on your own with the different effects. But I must say that this is also connected to a tight cooperation and a tight infrastructure in Germany. So we do have a lot of companies and a lot of academic institutions working on medicinal chemistry, working on drug development in the different areas. Just to show here, these are different companies you mostly know by name, or some of them you know by name, and they are all organized in research-based pharmaceutical companies localized in Germany with their research institute in Germany also. So there are some big institutes in this way. In fact, this research-based pharmaceutical organization has more than 870 study groups in Germany, and they have the production in Europe, Japan, and USA, and they also have the production of their drugs in India in this way. So they have, lots of them have also their production routes in India with tight connections to India. Yeah? So thereby, we do have a lot of companies involved in this. But these companies have a lot of experts, and where do the experts come from? They come from the academics. And you see that the students and the student education, PhD students, are quite important. And you see here that university students in mathematical, natural science, IT, and engineering in Germany, it's about 1/3 of all students in Germany. They are localized in these different MINT areas in this area and thereby. So thereby, this good education gives good benefit for industry, gives a large number of experts. A large number of experts gives them good drug design at the end. So you could not demand to have a good company or have a good company with outcome without having good education for the people. Yeah. And that is the reason why I'm here. That I would not give you a good education on medicinal chemistry. 1 week would not be enough. Maybe 2 semesters could be appropriate for an introduction. I just want to make you aware of the different problems, and that you get the awareness for self-learning in the different pockets in this way, and that you get some problems where they can work with different areas in this area, in the different structure. What we are doing in our educational program and our study program, that we also have societies for these different companies. So, for example, we have the German Chemical Society, and the German Chemical Society, they have a division for medicinal chemistry, and since a few years I'm one of the board directors of this section, and we are looking for the development of medicinal chemistry in Germany, in Europe, a little bit less in global terms in this way, because we are a German society and German organization and so on. And the same is true for the German Pharmaceutical Society, which has also section for pharmaceutical and medicinal chemistry. So we have 2 different societies connecting academics to industry people, and thereby enhancing the networking in the different levels in this way. Yeah? And they also enhance the educational processes. So, for example, they give travel grants, meeting supports, workshops, and what is, to my mind, most important, they give support for scientific networking. And scientific networking between academics, between industry, between biologists, between structural biologists, pharmacologists, and so on. So they support different workshops, different networks in this area, and so on. Yeah? Unfortunately for you, this is mostly German localized, because the Germans pay, they make the German support for the different areas, but nevertheless, they have this organizational step quite useful and quite helpful to my mind. I have seen that you do have a lot of pharmaceutical institutes here. You do have pharmaceutical academic institute or medicine chemistry academic institute, but I have not seen an Indian medicine chemistry society so far. Yeah? So maybe that would be a good starting point to create something like this. You have to start at a very low level with the beginning. The same was true with the different other societies here, but that gives you a good starting point for different associations and for improved networking in the different areas. Yeah? So that's maybe some point which is really important. These society then succeeded also in getting involved into publishing processes. So they are publishing scientific papers in this way, and they're running different journals like Chem, Chemistry, Nachrichtenchemie, Pharmakon, Archiv der Pharmazie, and so on, and so on, and so on. So there are really different possibilities in which this is on, and they are then also propagating their national research in this way. Right? You see like the ACS journal, they are propagating American, not only MAGA, but really in different areas in these sections where you're going on. So you see, it's also important that you get some inspiration, some motivation for the students also to publish in the different areas and to make your own journal. And to make your own journal as a high quality journal, not as a low quality journal. That's where you can go to some open access publishing processes where you can easily make up your own program or own journal within a few months. But here it goes for years, and thereby it needs years to grow up in this area. Yeah? I have been editor-in-chief for Archiv der Pharmazie, which is in fact the oldest journal of this. It was founded in 1822 in this way, so it has a long history in this way and growing up. Yeah? Not the most famous. The Angewandte Chemistry is the most famous journal in this way and has the highest reputation. So thereby, different possibilities where you're going. Now, this slide is a really important slide for you, because this slide gives you other teaching possibilities and gives you also the references for many of the citations or many of the pictures are used in these different lectures and so on. So thereby, take this as a reference for many of the structure where I did not put it into detail. I mostly tried my best to give the references in a good academic way, but due to time, I'm pretty aware that it was not always possible to do this in the full consideration, and thereby I recommend here to see this. One of this point is the European Federation of Medicinal Chemistry and Chemical Biology in this way. And you see the edition, it's called EFMC, and the EFMC is the European Federation of Medicinal Chemistry. It has recently changed its name into Medicinal Chemistry and Chemical Biology, because this is a production connecting both of them in the different areas and the different structures. And they do have a website where they have the called best practice, for example. And this best practice, I do not want to go here into this web-based structure. They do have some slides, they do have some graphics, and they also have some YouTube videos where they explain the different topics. And you will see there, for example, they worked on hit-to-lead validation or hit-to-lead development, validation of chemical probes, phenotypic drug discovery, hit generation, copyright problems, and so on. So many of the problems we are discussing here, and that was the reason I took many of these things here, and they are really excellent, I have to say. Yeah? So my congratulations to the authors of these. They have really done an excellent job, and they are freely available in this way. Yeah? To get access to the slides and so on, you just have to register, and that's possible. Yeah? And there's also a site for the Young Scientific Network, also there possible, and that's open for everybody, so even outside Europe could easily join this Young Network in the different pathways and so on. This respect, I would also make the announcement of the Next Generation Medicine Chemistry Group. I'm missing a R here, sorry for this. So the Next Generation MedChem Group is, for example, presenting case studies where people from industry or from academics develop some compounds or some candidates and so on, where they struggle with some problems, where they overcome the problems, and then in which way they succeeded and so on. And there they have quite nice things, so you can connect by LinkedIn or by MedChem cases. So they are the free lecture series. They are not stored, but they are only active during this lecture series and so on. But you can easily connect with them and get a lot of good experience from other people. And the best way to get your own experience, to learn from other mistakes and not to make the same mistake on your own. Yeah? So then you're much faster in the different ways. And I would also like to make some commercial applications here. Drug Hunter or Cambridge Medchem Consulting Agency, they also have very good possibilities. I also took some of their slides, which are open available for this discussion here. You will see this mostly in this way, but you can also register there and pay for additional information. So that's also quite helpful. Yeah? But I must say I do not have my money for all of the different consulting areas and all of the information series. But nevertheless, it's really an excellent information steps in this way. So all of the things I have here, I can recommend as excellent sources, and I also recommend as references to many of my slides. Yeah? Just to be fully clear in the different areas. So now we're going back, what is needed as a medicine chemist and where we're going. Yeah? Yeah. You see here, best practice is going, we have to know chemistry, biology, pharmacology, biochemistry, but also genetics, analytics, medicine, and so on. So medicine chemistry is a multifactional topic. It's not only chemistry and building up some targets, it's to know about the target, about the interaction of the target. It's biochemistry, it's structural biology and different aspects in this way. Yeah? So then you can develop some very nice compounds and get into the different ways. But having a nice compound, which is maybe highly selective, gives you just a chemical probe. And a chemical probe is not a drug. So the way from a chemical probe to getting into a drug, getting into market, is a different story and a different way to go there. Yeah? So to go translational, to go from your animal studies to human studies, to go from toxicological to human studies, to go from healthy volunteer screenings to patient screenings at the end. That's really a big translational process. Your precision, personalized medicine, precision medicine, personalized medicine, this really makes this really important. And at the end, you have to spend a lot of money for this development. And since this money can mostly be spent by pharmaceutical industry, at the end, they want to get the money back. So you want to earn some money in this way. And then you must calculate how much money I've spent for the development, how much money I will get back in return in a certain time scale. So the chief financial officer of your institute, of your pharmaceutical company, will tell you you can make progress with your project, or you have to stop the project. Even if you're successful, you have to stop because the money for investment does not display the return money in this different. Yeah? So at the end, it's a company who has to earn money. That's a problem. And with a stock exchange, you not have to make money in 10 or 20 years. With a stock exchange, you have to satisfy your customers each year to make some payments in the different areas. Yeah? So thereby, project management, time management is quite important in this way. And then it's also important to have some novelty, some innovation character. So if you work on a Me Too compound, it might work in some areas, but it does not give the great innovation in this way. Yeah? So thereby, innovation, novel cooperation, novel application, that makes a success at the end. So thereby, you have to go the ways others have not been going before. Yeah? And this makes this highly risky in different aspects, in different areas and so on. What I would like to stress is that you cannot make all of the different projects on your own. You have to team up. You have to get good partners, and you have to make the motivation of your partners really helpful and successful. And you can only have motivated partners if you're motivated on your own. So if you say, oh, I do this also on my own, and then I will work on this, and maybe next year I can give you some compounds, and then after that year, you have to wait another half a year, then the motivation is not really there. Yeah? So you have to be on fire for your project, and then you can bring it on for the next step in this way. So thereby, mostly there are some academic industrial collaborations, which at the end are the most successful ones. Yeah? But there are also academic collaborations, industry cooperation, and so on. So different possibilities. But you must also be aware that there are also sometimes academic industry competition in this way. So maybe you're not the only one who has recognized this innovative way, yeah, or this new way, and then there are high competition in this way. So thereby also be aware of your competitors in the different areas. Yeah? It's mostly useful to have consortia or multi-university partnerships and then get this knowledge base structural development into these different areas. Yeah? And thereby, sometimes pharmaceutical industry put a blame on the academics, which I, as an academic, cannot share for sure, but you will see that the academics sometimes is lower and has lower possibilities than the pharmaceutical industry, but a higher innovation character, or can work longer on a problem, whereas the money pressure in pharmaceutical industry would long have stopped such a project, and the academics, they have the stamina to overcome the different problems in the different areas. So thereby, it is highly successful, or can be highly successful, but it's not a company and another repetition of pharmaceutical industry. It's something different. And that's also the point where academics and pharmaceutical industry people speak the same language. They both speak Hindi, but they do not speak on the same level in this way. So they are talking about different things when they talk about being effective. The academic think that the drug should be effective. When the pharmaceutical industry think that they should be effective, the money spent for the project and the outcome, that should be effective in this way. So that's the main point where you have to differentiate between the different projects in this way. So thereby, this is quite important. I found a paper in Journal of Medicinal Chemistry recently last year where they said organic chemists should work here and know how to synthesize a compound, and a pharmacist who is more a drug specialist know what and why to synthesize the compound, and this together makes a good medicine chemist. Yeah? I must say it's a good starting point. Yeah? But to have really a good medicine chemistry or pharmaceutical development structure, you need much more than organic chemistry and pharmaceutical sciences in this way. Yeah? You see here, pharmaceutical medicine chemistry is a lot of chemistry, yeah? But chemistry also includes here the analytics, and analytics is just your NMR studies, your infrared, your purification of the compound, and then making some Raman spectroscopy, for example, with a compound, or fluorescence characterization and so on. But it's also biochemistry analytics. So going into the signaling mechanism. And then you go into biochemistry. From biochemistry, where you work on cells, then you go into organs or humans, and then you go into medicine. You look for the target, molecular biology, physiology, pathophysiology in this way, pharmacological aspects. You go into physics in a different way, and thereby I can stress the point from the introduction also into mathematics, into statistics in the different areas. So all of these together, this makes a good scientist in medicine chemistry. Yeah? And you see, it's a broad field having all of this, and thereby you need to team up. You cannot be expert in all of this, but you need to get the basic knowledge in all of this. As a good medicine chemistry, all of this is needed, and you have to speak to the experts in the different fields. You cannot be expert in all of these fields, and thereby this is quite important. And you see here, I took the IUPAC definition, the International Union of Pure and Applied Chemistry for medicine chemistry. It's a chemistry-based discipline involving aspects of biological, medical, and pharmaceutical sciences. is concerned with the invention, discovery, design, identification, preparation of biologically active compounds, the study of their metabolism, interpretation of their mode of action at the molecular level, and the construction of structure-activity relationship. That's the briefest definition I found for the way. So thereby, that's quite a point way. What you need is a bright mind and a bright idea. So thereby, you always look what could be get into the different things and in which way could it start. Yeah? And thereby, in many cases, you have a idea taken from a biochemistry sector and then translating it into organic chemistry. And then from organic chemistry, redirecting it to biochemistry or to biology into the different areas. So thereby, the different translatory processes and understanding are quite important for the different areas. Yeah? And if you ask the other, what do you want? Yeah? Thereby, I take the citation from Henry Ford. If I ask my customer what they want, they have said a faster horse. Yeah? And not a new invention of a car or something like this. So that's when you want something, you need something new, you have to think something new. Yeah? Yeah? And there's something special where you're looking for and so on. Yeah? In many cases, you can take the basic structure, nature, and you will have a lecture there special on natural compounds, explained into detail, where you can have the transferring from natural compound, for example, opium is still used in pain, mawang used as an asthma therapeuticum, belladonna or digitalis for heart failure and so on. So there are different starting points which are now in many cases overcome. So, but the nature gives us a good starting point for the different areas where we look for in the different stages and so on. Yeah? And thereby, if you have a good compound, then this compound must be transferred to a hit, that it can interact with a target and so on. And thereby, we go step by step this area that a compound had to be transferred to a hit had to be transferred to a lead compound then had to be transferred into a drug candidate. And then the drug candidate be transferred into a drug. You see there are step-by-step transferation processes, and being successful at one step being not is simultaneously being finally successful in the different way. But sometimes the intermediate point is also very successful and gives good academic record and so on. So not always the drug is really the most impressive thing in this way. Yeah? I took this, or I adopted this from a poet where Gertrude Stein in the 1920 explained a rose is not a rose is not a rose in this way. So thereby I transferred it into the compound which is not rosing anymore in this way. Yeah? So here you see development decisions in a different aspects from the aspect of pharmaceutical industry. So when you have a target selection, yeah, then you have to see in the medical profession, do they agree to this? Do the consumer advocates and the patient organization agrees that this is a useful target and so on? Do the regulatory agencies like NIH or something like this also agree that this is acceptable? Yeah? For example, if you would like now to develop some new opioids into the United States, you will not get any approval for this, yeah? Because they have enough problem with the opioid. So you have to use some non-opioids, for example, which have recently approved in this way. Yeah? And then drug hunter, market group, and especially here, financial analysts in this way. And at the end, you have to get the support of your research director. Without his agreement or her agreement, it would not work at the end. So thereby, you have to put all of the arguments together and then fight for your project, that this project is really enhanced and so on. Yeah? I know many projects which did not get this progression in this way. And then some of the researchers tried to get this project out of the company, and they bought the patent from the company for some use, and they made some startup company, and some really were successful. No? Not all of them, unfortunately, but you see there, sometimes the research director has not the best decision, but he or she will make the decision on their own in this way. And then the main purpose, what we have to decide if we have a good compound or a bad compound. We have a good drug or a bad drug into these different areas in this way. My problem or your? I'm getting radioactive, I think. No radioactivity, no leaves this room here. Is it working now? Yeah, it's working. It's working. How can you differentiate a good drug from a bad drug? Or in most cases, you do not have differentiated a good drug from a bad drug. In most cases, you have to differentiate a good drug from a better drug. So what is the purpose in this way? And thereby, three main aspects is the quality of effects. So what happens in the body really, what is there really happening? And this is not only target-oriented, it is in the whole body. It also concerns the side effects or the unwanted side effects. So that means all of this quality of effects are the wanted and the unwanted effects in combination. And then the quantity of the effect. So maybe if you go into pain regulation, that's also quite important in which level you get into pain regulation. It's tooth pain or bone pain. That's a totally different pain originations, and thereby normally you check the treated state to the untreated state into the different areas. And the other point is the duration. Yeah? Duration of the effect is also quite important. So if it takes from beginning to end, and is it beginning after 10 minutes or after 30 minutes or after half a day? And is the duration for 1 day or for 1 week? And if I stay with my example for pain, yeah, if you go for your dentist for some operation, then you do not want to wait half a day to get some pain relief in this way. You want to have it within the next 10 minutes in this way. Yeah? But if you have cancer and you have bone pain, then it doesn't matter if it takes now or half a day, but the duration should be quite as long. So thereby, it's not only pain treatment, it's what kind of pain, what kind of patient, what kind of origin of this we're really treating. And that makes me clear in which way we are organizing our targets and what we are looking for into the different aspects. That's the important point for the different structures and the areas we are looking for. We will have a closer look on the metabolic pathways of the different structures and of the different areas. And today and the next days of this week, we will go into further details of the different areas. And we have to reduce complexity. We cannot take quality of effect. What does it mean? Duration, what does it mean? Yeah? With the duration, we see that the absorption, distribution, metabolism, and elimination has large influence on the duration, on the effect and so on. And also with the effects are also maybe influenced by metabolites. Maybe metabolites have different pharmacological profiles and so on, and thereby this influences also the different effects. So thereby we look for the pharmacokinetics, which is the interaction of the body with the drug, and we look on the pharmacodynamics, which is the opposite, which is the drug interacts with the body. And thereby we have our receptor interaction, the mechanism, the different kind of interaction. So we reduce our complexity of the effect into different subgroups, into pharmacokinetic, pharmacodynamics. And even these effects are too complex, so we reduce these complex effects into smaller fields in this way. So it makes it easier for understanding, easier to handling, and easier to oversee in this way. Yeah? The same is done when you look on molecular modeling or different other aspects. So we use complexity and increase our understanding by this reduction. So we can look at the whole protein, or we can look at the peptide structure, or we can look on the localization of different important amino acids. And you see, if we look on the localization of different amino acids, it's much easier than to look on the whole protein in the environment. And thereby, we have to reduce our complexity. We have to maintain the problem, not change the problem with the reduction, and we have to reduce it as much as possible. Yeah? But as Albert Einstein said, you can reduce your problem as much as possible, but not more. Yeah? Sometimes people reduce it then to one atom or something like this. I think that's not workable. Sometimes you can reduce it to a charge or something like this. That's sometimes possible in this way. So thereby, understand also the problem, in which way this can be handled, and so on. And then with this understanding, then we can introduce this into drug profiling in this way. Never understand different drug profiles in this way. You see, today or in the first lecture, I will bring a broad overview on the different aspects we are then handling in detail in this way, and I cannot handle all of these things later on, but I will handle the most basic aspects there also later. And here we will later on look on solubility, look on metabolites, look on buffer solubility, maybe some metabolic stability, and so on. So we will handle different aspects in this way. Today I would like to increase your motivation just on the medicine chemistry project, on the truth of the medicine project. So in which way you get some hits, you get some hit to lead campaign or lead optimization into this way, where you get into this way. And that is more than just virtual screening. There, at the end, you have some wet screening at the example. You have to go into further details in which way you're going for the different structure. Yeah? So hit for which target? So de-risk your project. Yeah? So which target selection you're using? Which target is important? Which target is innovative? Yeah? And which screening system we are taking for this target? Does this screening system really give me insight on the disease we want to treat at the end? Yeah? And maybe you see, for example, GLP-1 agonists, the new peptide compound used for diabetes and now used for weight reduction, worldwide blockbusters, billion-dollar income into different areas. Novo Nordisk developed these compounds 10 years ago or 12 years ago and brought them into market as an anti-diabetic compound. And even they recognize the only with their own compound years later. And see what they have. So now they have, nevertheless, they are successful with this, but you see which way it's really important to see the omni page of all the aspects and the effects, not only for diabetes, but also for weight loss. And these Novo Nordisk was a company mostly working with insulin, mostly working with diabetes, and that was the reason all the people there were focusing on and almost forgetting. Yeah, so thereby you see in which way this is really important and can change the company really from this. And you see that with Novo Nordisk is a very good example because they were successful with their GLP agonists at the beginning. Yeah? So they got another diabetes therapy, but not really better than the other one. And now since they are hyped in this way, they're getting billions of dollars for income in this way. And they could not even succeed in giving enough supply for their compound. They missed a lot of income because they could not select their production line in this way. So thereby it's quite important into this way. So thereby we have different structure. And I will later on also go for assay development, in which way this makes some change and change in destruction. If we want to understand the different properties, then you see here with a easy cell cartoon, that this easy cell cartoon gives you some different compartments, gives you mitochondria, gives you cell architecture and so on, and gives you membranes, nucleus membranes, outer membranes and so on, different functions and so on. And then you have to understand this at the cellular level, in which way your compound is working. And if you understand this at the cellular level, then you can go to the tissue level. Then you can go to the whole body and so on. But without understanding your targets at the cellular level or at the structural level, I think you will not succeed then understanding much later. So thereby, it's important to understand and to have also good knowledge on biochemistry and biochemical interaction for the different structures. Yeah? I have taken this underground section to have this target scouting, proof of concept, clinical candidates, preclinical safety, clinical trials and so on, and then you can go into a marketed structure and phase 4 clinical trials and so on. So there are different steps which are occurring into these different areas which can be taken. And the most point when you, or when you want to take artificial intelligence or something like this, is the different screening areas. And you see, at the end, you have to be active in vivo with your patient. Yeah? Before that, you would try in vitro screening with, or in vivo screening with animals, and then in vitro screening in cell cultures and so on. And before that, you can have some in silico screening. But my main point here is that before all of this, you have to make up your own mind. Your own thinking, your own motivation, that it's worth putting your workload in this, putting money in this project and so on. So thereby, this in Cerebro is your own brain, and that's really the most important thing with this. It's a basis for all of the other things. So if you're not motivated for your own project, then you cannot motivate others. If you're not convinced that your project is the most important thing you can do in the world, then maybe you will not be successful. Yeah? And you can see with different startup companies, they're only successful if the people are really on fire for their company and for their project. Yeah? And I see as a PhD student, it's difficult to get some projects step by step, but I have to say, I started my own PhD project on the histamine. I was burning for this from the, not from the first year, but from the second year on, really. So when I get more familiar with this, when I learn a little bit more on this, and then we developed step-by-step further compounds in this way, and then we were really successful in cooperation with other highly potent partners in this way. Yeah? So thereby, that's quite important in this way. I'm not sure on the timetable how much time I have left. Okay, that's fine. That's fine. Because now I want to go into some further details with this. Yeah? And here I go into some details and some examples, and would explain on one example why this is important really for other purposes. My example, most of my examples go for G-protein coupled receptors, and there we had this rhodopsin-like structure or rhodopsin structure, which was the first G-protein-like receptor structure in the year 2000 or something like this. And then only in 2020, the G-protein coupled receptors were crystallized, and we got the real structure for this. Yeah? For example, also honored by the Nobel Prize for Brian Kobilka, so that was quite nice for this. Yeah? And then we do have the structure that we have 7 transmembrane domains. We have the G protein as a signaling transducer, and then we make the second messenger, which then activates different enzymes or different other structures and so on. So that's the main purpose in which way this receptor is activated or inactivated and so on. And then we do have this binding area here, and depending on the binding area, we have movement, some change in the transhelices structures, the transmembrane helices structures, and then the signaling is inhibited or induced or activated into the different steps in this way. This is a receptor, and thereby the receptor is a protein. Yeah? So you see this is a cartoon of a protein in the different areas. And the protein is quite... Maybe that's better. I'm not sure. Not really better. I try so, like this. Okay. Then, when we have a protein, this protein is always in flexibility. And this flexibility means that we have, for example, the receptor R fully inactive, and we have the receptor R star, which is fully active. Yeah? And thereby, this conformation status is flexible, and we have some flexibility where the totally inactive and the totally active receptor state would, with a short time scale, would occur. But we would also have some baseline where most of the receptor are some baseline here, which we call basic state or constitutive activity. This basic state or constitutive activity is not zero, because of the receptor are moving into the different steps. So baseline activity is a constitutive activity. We normally take as 0, but you can go below 0 because you can go from the basic state to the inactive state. And you can go from the basic state to the active state to R star. And this makes a difference into the receptor activation. So receptor activation could then activate this receptor at the full active state. And the receptor ligand interaction could also qualify that another state is a more stable structure in a dual element, and that you have some stability here, which is a partial agonist. Yeah? And then you can have an antagonist, which stabilizes only the basic state, and then with the basic state you would not see anything. So this is called a neutral antagonist or silent antagonist, because it does not change the basic state, and you do not see anything of this change in behavior and so on. And then we have the compounds which activates the fully inactive receptor, the red R in this way, and they are changing the state from the basic state into another new state. So they are changing like the agonist, the receptor, but changing it to the other side. So these are called inverse agonists. Inverse agonists are another name for antagonists because they are blocking the receptor. They are enhancing the inactive state in this way. And then you can have also partial inverse agonists or full inverse agonists in this way. So thereby, it's a clear behavior in which way these things are really different in different aspects in this way, and we have different compound efficacies. Agonist, full agonist, partial agonist, neutral agonist, or neutral antagonist, better, and the inverse agonist as another class of antagonist structure. Why is it important to have this distribution of the different areas? I show you here some dose response curves or concentration response curves in this way. So we have here histamine, and histamine is increasing the neurotransmitter level here, measured as arachidonic pathway liberation, and histamine and another agonist is emitted, and you see emitted from the curve has a little bit higher affinity, but is also a full agonist in this way. That was not me. Yeah, the full in this way. I can more easily do the downside here, and with the downside we have ciproxifan, FOB 5, and so on. And antagonists are clearly defined as inverse agonists because they are reducing it below below 0 in this way. Yeah? And you see that proxifan, this compound, behaves as a neutral antagonist and does not do anything in this way. So it has some behavior behavior in the different structure. Yeah? This makes it this neutral antagonist. If we have here increasing dosing of proxifan, it inhibits the activation by an agonist. This is an antagonist behavior and inhibits the activation of the antagonist, of the agonist. But it also inhibits the further inactivation of the other antagonist, the inverse agonist. So ciproxifan and the other compound, they also get reduced in this way. So it reduces the agonist behavior and the inverse agonist behavior due to a neutral behavior with the structure. So thereby this is really neutral in a partial way in the different structure. Now we're getting, it is clear more, yeah, so because now we're going into the more complicated behavior with the different structures here, because here once more we have the antagonist behavior, the agonist behavior. Antagonist is stopped by proxifan, and agonist behavior also stopped by proxifan, and then proxifan alone is a neutral antagonist in this way. This is here already measured with the telemethylhistamine transferase, so the metabolite of histamine in a cell culture. Now we go into a certain step then here to in vivo screening and different screening system with this proxifan. You see here for the first time the structure of the proxifan with the different areas. I explained a little bit the way for the arachidonic pathway, the histamine release, the N-methylhistamine transferase inhibitase, but also the G protein structure. There, this compound behaves as a neutral antagonist in the different properties. When we have different screening systems, for example, other pathways, then it behaves as a partial inverse agonist, or it behaves as a partial agonist in different pathways. When we look, for example, for the monophosphate inhibition in CH cells, in Chinese hamster virus cells, it behaves as partial agonist. And further on, when we go then not only to the functional, to in vivo screenings, then we do have really some more. So if we look, for example, sleep-wake state in mice, it is an inverse agonist, so it induces. But when we have here in agonist, and then it is a full opposite behavior in this way. Yeah? And in fact, when we then go for the cat, it also makes the cat awaken, so it is a full agonist with the cat. Why does it come so different here? Huh?
Can you please give an example? Differentiate between neutral antagonist?
I will explain it because here with proxifan, here is a neutral. We have explained proxifan as a neutral antagonist. Yeah? In our assay for arachidonic pathway and for telemethylhistamine level. But here you have to see that this compound here, that with our different structures, the inactive and the fully active structure, and we have the basal state. And the basal state, the structure where we have this basal state is not always the same basal state. This condition, the zero condition, is different between different species, and you see it's fully different. It was agonist to antagonist behavior, because the cat is highly active during daytime, and when you experience the mice during daytime, it's their normal sleeping time. So their basic state is very different. And when you look for different diseases, the basic state is also there different in this way. And when you look here, when we have different basic states to different areas, and then we stabilize a certain step in this way, some compounds work as agonist, because when we have a low basic state, then the partial agonist is the more active state. But when we have a high active basic state, and then we stabilize the same status, then we have a partial inverse agonist behavior. So we go below the basic state. When we have a high basic state, then our compound is an inverse agonist. When we have a low basic state, the same compounds behave as a agonist. And this should make you clear that even pharmaceutical definitions are communication. It's not the property of the compound by its own, it's the property of the compound in connection. So it is the communication you're looking for. So efficacy or affinity or any other property is not the basic property of the compound, is the interaction compound with the target. And sometimes the target with the mouse is a little bit different of other things because of some species relevance. And especially when you look for pain, the great difference if you look for always pain to have sometimes, because they have different basic states in the pain level and so on. So thereby this communication, if you take this as a first take-home message from the first lecture, then I'm fully satisfied with everything, because then you see the relative properties of these different compounds and of the different effects in this way. Yeah? So thereby I will not stop here, but I will mini material. Maybe I cannot go here into detail for all of this, but you see the IOPHA, the International Union of Pharmacology, and the British Pharmaceutical Society, they do have a lot of pharmacological databases. They, the Drug Bank, Drug Central, and so on, they do have a lot of different properties where you get informed and where you have a lot of possibilities, where you get a lot of drug information, drug-related information, target information, and so on, the different properties and so on. Yeah? And I also can support the FDA Orange Book, where patents are published. So patents are protection of intellectual priority. So they do not give their information quite easily. They want to protect the information, but nevertheless, they this information at a certain level, at a certain time scale into this way. Yeah? There are further information for patents, for molecular database, for isosteres, bioisosteres, G-protein coupled receptors, and so on. And you hopefully see that I cannot go into details with all of these things and all of the structure. But if you access some of our web bases and so on, or Drug Hunter here so on, you will easily go all these different web pages and so on, and they are connected with Hunter for information on this way. And you also see that there are a lot of tools for the prediction of the pharmacokinetic properties and so on. Some of these are free, some of them are free for the academics, but most of them are have to be paid. So most of them are quite commercial in a way. So pay attention when you use these, which are really had to be paid and on a long-term basis, or which can be used for free for the different aspects in this way. Yeah? I would more or less stop here with this structure here we're having here. And with this structure, there was some information, some discussion with people from pharmaceutical industry. And with the discovery plan, you go from drug screening, hit to lead optimization, in vitro optimization, lead dynamic studies, and so on. So then even go to manufacturing, and go to in vivo studies with humans, so with the clinical trials and so on. So thereby, it's a red line you have to use here, yeah? But you see the red line is not always straightforward. Yeah? And it's more or less like this red line you're having here. Yeah? So in which way you can transfer your mouse model into the human disease model? Which way does this really reflect? Say comparable to another assay system in these different ways. In which way can have toxicity? Is a toxicity based by your target or based by your compound? you always have some kind of toxicity, so a very rare toxicity way, and thereby you have to explain what kind of toxicity is. Is it compound related or target related? If the toxicity is target related, you can forget your target, because you will always have toxicity when you address this target. Maybe some other addressing mode would be helpful, but nevertheless, thereby the reliability is quite important. And then when you have some animal screenings with some dosing and you treat correctly, in which dose you go then to humans, which is it transferring the predictable dose for humans. If you can try one, two, maybe three different doses, but if you fail with this, you fail with your whole experiment in this way, because you go only into human once. So it's important. So there are a lot of obstacles, and thereby all these obstacles have to be solved. And one recommendation is don't work on the things that don't work. It's easy to say, but at the end, you have to work on the things that do not work and have to make them work. That's innovation. That's the character. Up too early, but on the problems who are solvable for you. So that's something you have to go in between and then see in which way this is going on. I promised a little bit too much. That's not only my last slide, but here for some areas for distribution. So we only work on small molecules. The overcome from small molecules to biologicals quite easy. Peptide synthesizer, so when you have small molecules or when you have a peptide or a small molecule going, so thereby the transferation from a nucleotide or a polypeptide into a small molecule or not small molecule is not always as simple as you say. There are some green areas in this way, and we mostly connect to the small molecules which are by organic chemistry and not by synthesizers by them own in different steps in different ways. So that's what we are working on in the different areas. I will not go into the different phases of clinical development. I will not go into phase 1, phase 2, phase 3 clinical development. I will only stick with the preclinical development, because this is the major opportunity where the chemistry can interact with the different properties. So when you develop some new compounds, you can develop some new possibilities and so on, and so on, and so on. I will go slightly for some other points, but normally here our preclinical discovery process is that what we are focusing on and what we are looking for. And then what we want to have is innovation at the end. Innovation means a new medical entity. New medical entity could be a new therapeutic entity, new molecular entity, new biological, new chemical, or new active compound. So there are different definitions what is new. And thereby this novelty and innovation character is quite important for the different purposes. So Sir James Black, who received the Nobel Prize on the beta blocker and H2 receptor ligands, he made a very important stress. The best point to develop a new compound is to start with an old one. So if you have an old one and you have recognized some side effects, then change your compound in this way. So improve some old therapeutics, develop some new lead structures with less unwanted side effects, a new mode of action, or drugs for new targets, and so on. So that is mainly for drug development and so on. But with a drug, you should develop a disease treatment. And that's then quite important for which diseases you should select in this way. My recommendation is to select innovative drugs with the diseases with a high social burden, cancer, neurodegenerative diseases, input on the whole society worldwide. We have some diseases with high medical need. We do have the appropriate treatment, or for example with the antibiotic, we have the need for actual innovation with these different structures. We do have some diseases only with symptomatic treatment, and there we need a causal treatment for this compound. And also everywhere we have some unsatisfying treatment options this way. So thereby I clearly see that medicinal chemistry should be help for the people, should be help for the society, should help giving drugs for a better society into this way. Yeah? And thereby your natural sciences, but with this, your global, you see that the problems with the antibiotics, they're getting stronger and stronger because of the resistance of numerous antibiotics. So new antibiotics are strictly needed for this purpose. And thereby, I would like to stay with this slide as the last point here, because this is the point for your process. And then we will go with the physical-chemical characterization, some other structures later on after lunch. Yeah? Thank you very much.
Thank you so much for this lecture, sir. If anybody has any questions, you can ask.
Don't be shy, we are fully open here. We are.
You give some separate award for the question and
Over there?
Yeah, thank you, Professor. Very nice talk. So I have a question related to purity of drugs which are administered in the body. If the purity is less or more or not 99%, so what is the effect of that on body? And there is one concept you said about toxicity of the drug when it administered because of drug. But before administering drug, so whatever drug formulation is there, that is having some toxic impurities. So how can you separate that? What is the role of organic chemistry into that? And if it is there, so then how it affect addition to drug toxicity, how those impurity affects?
The problem for anti-herbal drugs and so on, so you want the toxicity of this compound for this kind of, but then you must avoid any human toxicity. So your then should be on fungal, on plant cells, but also simultaneously on human cells. And then they should have only this toxicity effect on the non-human cells and should have no toxicity effect on the human cells. Another main problem there is that some of these compounds are metabolized, and with the metabolization you get some new compounds, new pharmacological profile. Then it's not only satisfying to have the pharmacological profile of a compound on its own, but you also need the pharmacological profile of the metabolite. And then you have to synthesize the metabolite by organic chemistry, and then screen the metabolite on the it's the only way you can go for this before you go into we have to clarify the toxicity profile is appropriate for the species have taken there must be clear also much longer before we go into humans or any other trial. So toxicity is the main problem, and we will see later on there's a large cascade what must be done in the toxicity profiling for the different compounds, what had to be ruled out in channel activity like hERG activity and other activity where you're not only going for gastrointestinal interactions, but also going for cardiac infection or for brain interaction, for fetal interaction. So that's quite a large number which had to be checked. For toxicity, the main purposes are before you're going into human trials, all of the vital reaction must be performed. So CNS, breathing, and cardiac function had to be checked extensively. That's the priority.
Thank you. Thank you.
Thank you, Professor Stark. I do not really have a question at this point, but I have a feedback to give. Your presentation was very excellent in setting the base for this course. You have touched almost every aspect in your in the beginning, which students and all the participants should know, and now they can expect what is coming up. And I really appreciate the diagrams which you have put in your slides. They were very interesting. We don't come across those diagrams in the internet, especially the one explaining the drug discovery process, including the drug development process, because all of us are so bored of all the diagrams which are available on the internet. So many thanks for setting up this tone for the course.
Thank you very much.
Do we have any more questions?
Hello, sir. When we are having a targeted drug design and we are targeting a specific biomaterial, so what should be our target? That whether how my molecule is binding with that biomaterial or the transfer process like carrier proteins and all. So and how much screening is sufficient?
Yeah. There are different possibilities to define your target. Sometimes some kind of cells could be your target. Yeah? If you're looking for bacteria or something like this, then the whole bacteria or membrane or specific enzymes could be the target. And that's not really clarified if you just have to bind it or if you have to activate or deactivate it. And you will learn, I think on Thursday we will have a lecture on diagnostic agents, so they will destroy your target in a way, in a different. So there are different possibilities in which this could be done. And, for example, anti-schizophrenic compounds, they all bind to the same receptor, and they inhibit the receptor to the same extent. But the different anti-schizophrenic drugs have different clinical outcome because of the kinetic of the binding. So how long do they inhibit the receptor? So in some cases, it doesn't matter for the schizophrenia, the neurotoxicity, it matters quite a lot. They are having then different properties. So their affinity is not only the only part for evaluating the compounds, they're the on-off phase for the receptor are another important point where you have to struggle. When you look for kinase inhibitors for cancer, then selectivity gives an advantage that you can exactly inhibit only one kind of cancer. But this kind of kinase, sometimes this selectivity is not wanted because you do not have one specific kind of cancer, but different cancer. Thereby, the non-so-specific kinase inhibitors are slightly more advantaged than highly selective ones, because the response of the patient is much better. And then you can have the inhibitors, and the inhibitors can bind to the kinase in the open phase, in the closed phase, or another phase, and the newly developed also described here, will have some covalent inhibition. So they bind on a thereby the activity of the kinase, and the kinase had to be rebuilt in this way. So depending on your disease, the kinds of action which are the best ones, and I can the best in which way this is optimized. And you see with the kinase inhibitor, different they were not started as an ATP competitor. They were to be probable competitive inhibitors, less selective compounds. There are different kinds of knowledge in which we can. So, unfortunately, I cannot give you this kind of interaction. So, the best kind of interaction is to treat the disease. And there you have a number of different diseases with different receptor interaction. Maybe I can. The areas. Compounds which have to be activated. These superagonists do activation of the receptor, and the activation of the receptor the signaling mechanism, thereby the superagonist were functional. So, and now some functional antagonist as a new compound, mechanism of action, prodrug activation, phosphorylation. So this mode of action was optimized with some higher receptor activity. But you see, for this, in this case it was multiple sclerosis in this way, so that's easily and you understand your receptor system and your receptor interactivity.
Thank you, sir.
But very good question.
Good afternoon, sir. You talked about different ways like in vitro, in vivo, in silico, and last one was in cerebro. I didn't actually get what is in cerebro. Okay.
Own brain, that's the most important point you have to work with, and you have to think about it. You interpret the data. The data are just data points, and in which way you are taking this as a new inspiration or as a new possibility. For example, we are looking for, and we the profile of new drugs. There we look at the compounds new developed, side effects detected, or compound in clinical. Maybe some compounds which have we go into detail the brain, what could be the, and then after that we can take some in silico screening or some other things on. But I think intellect to work with and your own broad expertise in the different fields. Without this, you get lost. It's like when you're using some videos on TikTok. Yeah? You have TikTok or something like that. There are sometimes, or it's getting more differentiate. So you have to use your brain always priority or some artificial intelligence to check this. In most cases, they are artificial intelligence, and thereby you have to use your things. Or when you get some offer by an email, you can get a new iPhone for 10 euro. You can also be aware that this is a fake. Well, you do not have to make some screenings on this. Just take your brain in this way. That works best.
Thank you, sir.
What extent in-cerebro research has supported your research?
In-cerebro has largely supported my research because we are some of the compounds development we are started at the very first beginning. The histamine H3 receptor where we worked on was just newly developed, and we were in connection with a person who has recognized the receptor, and thereby we were able to check the compounds. Firstly, we worked on agonists. I take it in my hands, maybe that. This is the same. Firstly, we worked on agonists, and we were not sure because it's a Firstly, we worked on agonists because it's a presynaptic receptor, and we hope with agonists we got a negative coupling in this way, and thereby got some antagonist behavior in this way. And we prepared compounds based on histamine with methylation or with other modification and so on. And they showed out to be not effective in the in vivo screenings we or our partners have used. And then we made antagonists. And our starting point is that we took the agonist, so we took an old drug, the endogenous ligand, and then elongated this compound. And by the elongation, we maintained the affinity, but we changed the efficacy. So we maintained the affinity by the histamine binding core, but due to the longer compound in the way, we stopped that the G protein coupled receptor changed its position. It maintained in the original position, and thereby we had some inverse agonists and neutral antagonists in the way. So thereby no other antagonists were known, and then we started by the agonist and changed it by another way. And in fact, this is not new, because if you look for beta blockers or so on, you use dopamine, and then you make some elongation in this way. It's an old story, and in fact, we had the benefit to work together for a while with James Black, who had developed this, and Robin Ganelin, who developed cimetidine, the first H2 receptor blocker. So it's an old trick for a new compound.
Thank you.
Good afternoon, sir.
Yeah.
Sir, my question is, what are orthosteric and allosteric ligands and how they work?
Yeah. The orthosteric ligands, they are ligands which have the same binding site as the natural agonists. So orthosteric means the same binding site on the receptor, on the enzyme, or so on. The allosteric means, from the Latin translation, another binding site, another area, so outside of the natural binding site of the endogenous ligands in this way. And they can have some other properties, and they influence the activity or the binding site because they are changing the position of the different structure. It's difficult to say. There are many allosteric modulators who change the binding site. Most allosteric modulators are so-called negative allosteric modulators, so they change the binding site that you have a worse binding. Yeah? but there are also some possibilities for positive allosteric binders, and they open up the binding site so that the incoming of the agonist or the substrate could be faster. And then, for example, with a positive allosteric enzyme inducer, the enzyme got higher substrate transformation. So there are modulation not on the active side of the receptor of the enzyme, but on the other side of this. And there are also different possibilities, like we have here with the agonist or antagonist behavior. Also with the allosteric point, there are also some definitions for positive or negative allosteric modulation, but that's really beyond the regular binding site and thereby difficult to proceed. But at the moment, many people work with the molecular modeling procedure to find such allosteric binding site because they are independent from the open or closed conditions and thereby maybe more attractive for an enzyme or for a protein because the enzyme had not to be activated or deactivated and so on. So it's a generally activity possibility.
Thank you.
Oh. Behind you?
Hello, good afternoon. Thank you for the excellent lecture. So the circadian rhythm, the human in human body, the morning evening cycle affects chemicals differently. The circadian rhythm is the concept in biology, sir. They tell us that the homeostasis that maintained in the body changes according to day and nights. Means the human mechanism may differ according to day or night. Sir, what are the expected results of circadian rhythm on the drug development? Because the drug is the same. Give the patient at morning and night, the drug may not differ, but the condition of the human body may differ, sir. According to that, is there any changes that we should consider before drug development?
You're considering here the circadian rhythm, so day-night rhythm, with the different drugs in the area. Yeah? And is this correct? No?
Sorry, sir.
You're concerning for the circadian rhythm.
Yes, sir. Sure,
yeah.
sure, sir.
Yeah.
Exactly, sir.
Yeah. And that's one aspect. This is highly underdeveloped with many drugs. For example, it is good developed with sleep and wake drugs, for example. But that's the original target you're working on this. But it has only been investigated during the last decade, for example, for pain. So pain reception during daytime and pain reception during nighttime in this way. And these individual problems, they are getting more and more into awareness into the different structures. But the same is true that there are also differences between different sexes. So male and female, they have a different responsiveness to pain or to other things in this way. They have different hormone regulations and so on. So the difference between the individuals, between different groups, male and female, between children and elder people, and so on, and between nighttime and daytime, but mostly between healthy state and unhealthy state. Yeah? For example, we have developed some compounds against drug abuse, and they were mostly effective against cocaine abuse. So we have, some of our partners have developed this compound for cocaine abuse in rodents, and even cocaine abuse in monkeys. Yeah? And they were effective in a certain dosage level. When we increase the dosage level, they were totally blocking, they lose their selectivity. When we are dosing too low, they were not effective. And thereby, we stopped this project at this phase because it was fully imaginative that we will get the correct dose with the cocaine abuser or to find the correct dose with the cocaine abuser, because these are not the most useful patients to follow up all conditions in this way. Yeah? So just to be aware, we were successful in compound development, we were successful in dosing, but we were not successful in the translatory process, transferring our animal data to patient data, because our patients were not as responsive as we done. So in real data, we could not transform our compound. And this is the same with the circadian rhythm. Not always all the data are transferable in this way. You do the best, and if they do some very strong side effects, you have to be aware of this, you have to look for this. But if they are acceptable in a certain way, which is for most of the drugs, you do not looking for the daytime rhythm. One example where you have to look for a daytime rhythm, which is very popular but not very popular in the disease, when you take testosterone or some hormonal treatment with male hormones, then you should treat it 1 or 2 hours after breakfast, because then your level increases in the way, and then you simulate your normal physiological circadian rhythm. And when people take this for the night, it's not recommendable, for example. So there are some recommendations in this way, but not always fulfilled in daily practice.
Thank you, sir.
It seems time for lunch.
So is there any more questions? I think they have all understood. So anyways, excellent lecture by Professor Stark. It's a fast session, was very nice. So I think now we can stop these sessions, and you remain seat. We'll be going for lunch very soon. Okay. So then we'll rejoin the next session after the lunch. Thank you. 2:30 around. Maybe how much time? So we have sufficient time. Okay. Thank you.
Hello.